invite the honorable mention abstracts panel to come up to the virtual dais here. We were really blessed with amazing quality of abstracts that were submitted. Not everyone can be a prize winner, but we have terrific honorable mentions that we'd like to have presented in this session. Yes, I should introduce our panel here. We have Georgina Xanthou. She is a visiting professor of immunology at UCLA. And we have Alex Klonoff. He's an MD, MBA. Both of them have actually joined Kinexum within the past half year or so, but they've been phenomenal, indispensable in helping us to organize Metabesity. And they had among a particularly material role to play in the abstract solicitation. So let me get out of the way and pass the mic on to you.
All right, welcome everyone to the abstract honorable mention presentation session. I'm Alex Klonoff. With me is Georgina Xanthou. And so we're going to start with Kelvin Ying. Well, first, I guess before we go into Kelvin's presentation, we just wanted to thank everybody for their abstract submissions. Everyone had really amazing science that they've been presenting, and we appreciate the submissions, and we're excited to read through all of them, and we sent them to our abstract selection committee. And so these presentations were not selected as the finalist awards, but they were very excellent, and we wanted to have them showcased at our conference as presentations. So the first abstract will be from Kelvin Ying, and when you guys present, if you can also state your affiliation with which lab you're in, that would help us with the conference to know more about what you're doing.
Hello, everybody. My name is Kelvin Ying. I'm in the lab of Dr. Huixun here at University of Southern California, Leonard Davis School of Gerontology. Today, I'm going to present my newest data on MENSH, novel mitochondrial microprotein that is involved in diabetes. And just a little conflict of interest statement that USC makes me say is that USC, myself, and Huixun have IP rights, and we have a patent for MENSH and the analogs that I'm presenting here today. So just a quick background. Of course, diabetes is a huge problem in the United States and the world in general. Over 10% of the population in the USA has diabetes. 50% of people over the age of 65 have prediabetes already. And in the US, it's the 7th leading cause of death, although some can argue that it's the 3rd cause of death, and which will become, this information will be, you'll know why I'm saying this later on, but the incidence is different among different ethnic groups. And in Mexico specifically, it's actually responsible for about 1/3 of all deaths of people between the ages of 35 and 74. So my lab where I'm working with Dr. Huixun, we focus on the mitochondria. And so we were looking at the connection between diabetes and the mitochondria. There's a lot of different connections between there, but I mean, diabetes itself can be caused by mutations in mitochondrial DNA, and mitochondrial dysfunction is found very commonly in diabetic patients. And so our lab actually focuses on not just the mitochondria, but very specifically on mitochondrial-derived microproteins. And so you can see here in the diagram here, it's actually these microproteins, which are defined as proteins under 100 amino acids, are encoded by small alternative open reading frames actually within the mitochondrial DNA itself. And they're found circulating throughout the body, plasma, CSF, other tissues. And in general, they act as endocrine signals, but they can also work paracrine signals or just even internally within the cell itself. And so here we have the first mitochondrial-derived microprotein called humanin, and it's actually found within the 16S rRNA gene of the mitochondrial genome. So it's a kind of a gene within a gene, a nested, like a Russian doll type of situation, kind of. So the question that we were wondering was basically, can mitochondrial DNA variations explain some of the differences in diabetes incidences between different ethnicities and races and things? And so what we did was we took a study, a genetic forward approach, where we did, instead of a GWAS, we focused just on the mitochondrial genome so that we don't have such a huge threshold to get over when you look at the whole, human DNA, right? It's a much smaller, it's only 1,600 base pairs versus, the giantness of the nuclear genome. But what we were able to do is we found a mitochondrial SNP that associated with increased hemoglobin A1C levels, which indicate uncontrolled glucose levels, basically. And so the A and G are just two different SNPs or variants within the mitochondrial genome. And you can see here that if you have this alternative allele, G here, although when you're normal weight, it doesn't have any effect. As soon as you're overweight or obese, you can see that you have this great increase in A1C levels, indicating that you probably have diabetes at this point. And so interesting, because it's mitochondrial DNA and mitochondrial DNA are passed only by, is maternally inherited, you can sometimes figure out what regions and ethnicities carry this specific SNP. And if you look here in the green, it's people that actually carry this alternative version. And you can see it's mainly it's actually kind of a Native American specific SNP. And so when we looked, because we were interested in microproteins, we looked at the SNP to see what could be going on. You know, if maybe it's the larger gene or the smaller internal gene that has a change in amino acid sequence. And we found that actually, so this SNP is within the MTND2 gene, and it's a synonymous mutation in terms of MTND2. So there's no amino acid change here. But when you look at the small open reading frames that could be there, we found that it actually interrupts the start codon of this microprotein that we called MENSH, actually. So people who carry the SNP are theoretically knockouts and do not produce this microprotein at all. So this is all very good and well and theoretical, and it's possibly a microprotein. So what we did, and I don't show this data, but was we had synthesized this microprotein, threw it onto cells, fat cells, different types of cells, so in vitro work, and if we found that it did have some biological activity. And then what we did was then synthesize some more and then actually put it into mice to see that if we can replicate what we would think would happen with this SNP in humans, right? Which is to say that can this new microprotein MENSH help prevent some diabetic phenotypes. And so we put mice on a high-fat diet, which is, kind of pushes them towards diabetes. And here you can see that the control mice, and we gave them a glucose tolerance test to see how quickly they can get rid of the glucose. And if they actually have diabetes, they usually have a worse ability to take out the glucose. And so here we injected mice actually twice a day or just one time a day with a high dose. But either way, we administered it IP and injected it. You can see that when you inject MENSH, you actually have an improvement in glucose removal from the system, basically. And then this is just the quantification here, but it just shows that when you do inject this MENSH microprotein, you do have improvements in glucose tolerance. And then we also made a lot of analogs and mutated it, because actually this microprotein is only 18 amino acids long. So you can actually go and mutate every single amino acid residue and look for different biological effects, and when we're messing with the structure, and do all sorts of kind of fun things in a kind of a straightforward way. And so you can see here that we put mice once again on a high-fat diet, but when we had MENSH 32, which is 32nd analog that we happened to synthesize, we were able to actually completely prevent weight gain on a high-fat diet. And these mice actually were eating just as much food as their control counterparts. We just used another model of diabetes, which in this case was the OB mouse, and these mice don't produce leptin. And you can see here in the lower right corner an OB mouse, which is grossly obese and diabetic as well. But even with this huge increase in body weight and diabetes, with our MENSH 32 analog, we were able to prevent this weight gain. We basically cut it in half. And here you can see it is actually largely due to changes in body fat. And so this time we were actually aware that, that we were going to see some nice changes in body weight and other parameters, and we put them in a metabolic cage. And here you can see just the metabolic cage, their respiratory exchange rate, or basically metabolic rate throughout the day. And you can see this in the circadian rhythm. But what you see here is the control. And then here is the mice injected with MENSH 32 MENSH analog. And when you go down to about 0.7, it means that you're utilizing pure fats. And then 1 is usually indicative of you using pure carbohydrates. And you can see for reasons that we still don't know, but we're trying to figure out, you have the switch over to metabolizing fat over carbohydrates in these mice. So of course, we were trying to look for some mechanism, what's going on here, how does this work, right? That's everyone's big question, of course. So we looked at just the obvious candidates, which in case, is the AKD signaling, which is directly below the insulin receptor, basically. And so what we found was basically that when you add mensch to these mice and look at muscle and fat, you can see that there's a differential effect within the insulin AKT signaling system. So this is just the AKT 308 site and the 473 site, but you have the same data where you have a differential effect within the muscle and the fat of these mice. So that kind of gives a little bit of a hint of what the mechanism is, but clearly we have a lot more work to do. So in conclusion, we think that mensch and its analogs, it prevents weight gain, some metabolic dysfunction in different mouse models of diabetes. And we've done a lot more work than what I presented, but there's only so much time I can use to talk. And because this microprotein is actually absent in certain Native Americans and Mexican populations, this could end up being maybe like a first-line drug of choice for these populations. And one of the first examples would be, of a precision medicine approach to diabetes, which is not there yet. So,
thank you so much. Very interesting work.
So, yeah, very exciting data. Congratulations. So, I have a couple of questions. So, first question would be, so I have one question. Well, due to time. What exactly do you expect this protein to be doing? Is it also affecting some genes or the acetylation phases in the nucleus? Do you think it would be affecting some other proteins as well?
Yes, so definitely we think it's like similar to other microproteins that are signaling molecules. So we have been kind of trying to search for the receptor, right? Which is always a very difficult type of thing to do. So we have been trying, but yeah, and we've done RNA-seq data, so we can definitely show that there's signaling changes that affect the nucleus that end up, with your typical cascades and changing of nuclear gene expression. So.
Okay, and what about some other metabolites? Have you done any metabolomic studies to see if it affects the whole metabolome of the cells?
We did. We checked the metabolism, metabolites of mice on a high-fat diet and the plasma in particular, and we do actually, which corresponds with the OBOB metabolic chamber data. We do see more utilization of fatty acids and things like that.
So that's important to proceed to pursue in future
to go
studies.
That way. Yes.
Yeah. Okay. Yeah. Very nice. Very interesting study.
Thank you.
Thank you so much. All right. So next up, we have Albert Ying. And so he'll be presenting, and just let us know your lab as well. And we'll go through the presentation, and just due to time, so it'll be a 5-minute presentation. We'll do the Q&A after each of the remaining abstracts. Take it away, Albert.
So good afternoon, everyone. I'm Albert Ying from Harvard Medical School Gladys Shift Lab. And today I'm excited to share with you our new work where we build a first causal biomarker of aging, and we use it to separate the age-related damage and adaptation. So there are many research on aging epigenome, but most of them are correlation studies. It means that they study how the epigenetic mark change with age. But we don't understand the meaning of this change, like how would this change affect our lifespan and health span. This is because the age-related change is just one piece of puzzle, and we also need to know the causal consequence of the epigenetic change and to complete the picture and understand the aging epigenome. So, but how do we estimate the causal effect? The gold standard way is the randomized control trial. So basically, you can basically do a trial and separate people into intervention and control arm. You do the intervention to change the methylation level, and you observe their effect on the age-related phenotype. And this is the causal effect estimate. It sounds simple, but it's a huge problem because there are millions of methylation sites across the genome, and we're talking about doing millions of the clinical trial simultaneously, and which is impossible. And luckily, we have another approach called mediator randomization, which uses the same principle of the randomized control trial. And using this method, we can estimate the same causal effect of the methylation site on the aging-related phenotype. So here we perform a large-scale epigenome-wide molecular randomization analysis across 400,000 CpG sites across the genome, and to estimate their causal effect on 12 different aging-related traits, including 8 lifespan health span-related traits and 4 epigenetic aging traits. So here I want to focus this aging GIP1 because it's a genetic principal component of basically all other lifespan health span-related traits, and therefore can be considered as a genetic representation of the healthy longevity. And this is what the real data looks like. So we identified thousands of CpG sites across the genome that's potentially causal to healthy longevity, and they affect aging through maybe different mechanisms. For example, our top hit is on 4th chromosome, and it's related to the bone density. And another hit is on the, for example, this one is related to Alzheimer's disease, and there are so on and so on. And now imagine if we put the causal effect we just identified on the x-axis and the age-related change on the y-axis, we can actually separate the damaging and adaptive change. So for example, if you have a methylation site which promotes longevity and its methylation level increase during aging, this is actually can be defined as a protective increase. And similarly, we can define the deleterious increase, protective decrease, and deleterious decrease. And in reality, we found a lot of CpG sites that falls into each of this category, and surprisingly, they're roughly balanced. This means that there are similar amount of adaptive or protective change and damaging change during aging. And use this information, we built 3 different causal clock models. So the cause age, which is built using all the causal CpG sites we identified. The damage, we're using only the damaging causal CpG site, and the adaptage using only the protective CpG site. And if you just look at their accuracy on predicting aging, they are quite similar. So the damage and adaptage, they both predict age quite well. This is it's reasonable because both damage and adaptation against the damage are accumulated during aging. So both of them should predict the age well. However, when we apply this to clock on the mortality data, we found that people with higher damage are more likely to die earlier. So basically they have higher mortality risk, but people with higher adaptage are actually have lower mortality risk. So they are not, basically they tend to have longer lifespan. And this may be because they obtain more protective adaptation. And another example I'm going to give you is cellular reprogramming, which is a process that can reverse differentiated cell into a stem cell-like state. And this finding won a Nobel Prize in 2013. And recently, several studies shows that this reprogramming process may also be able to reverse the age of cell and remove the damage. And we applied our clock.
Sorry to interrupt. You have about 1 minute left. Just letting you know.
Yeah, I just have one more slide. Yeah.
Sure, continue.
And we apply our causal clock on reprogramming, and we show that the red line, the damage is decreased, is reversed during the reprogramming, while the adaptage is even slightly increased. This suggests that the reprogramming process is actively removing the damaging accumulation during aging while fortifying the protective adaptation. And this last one example I'm going to give you is a short-term intervention. Patient are given 6-week omega-3 supplementation, and you can see the damage can already show the effect. They slightly decrease their age, while adaptage are slightly increased. While this effect cannot be observed by all other clocks. So just to summarize, in this study, we identified the causal DNA methylation site for healthy longevity across the genome. We built the first causal biomarker for aging called damage and adaptage. And only damage are reversed during reprogramming. And this is our preprint. And for acknowledgement, I need to thank the people in GladyShift Lab, especially Vadim, Andrew, and Hannah, and also our fantastic collaborators. And I will take questions. Thank you.
Thank you so much.
So you want to go first?
No, you can take it away. Yeah.
Okay, so a question that I had is, are immune response gene affected either in the positive way or a negative way, either like the damage response or the protective response?
So here we focus on basically the methylation side. So we didn't look at how this methylation side could change the gene expression. So so their study shows that the methylation as promoter or enhancer could change the neighboring gene expression. So it's a possible like mechanism. Yeah.
Yeah. Okay.
My question is, were you able to incorporate any strategies to reduce potential pleiotropic effects of the techniques?
Yes, we applied basically the first thing we did is that we only use when instrumenting the CPG side, we only use the genetic variant like near the CPG site, basically in the cis region. And this will reduce some pleiotropic effect. And we also use some mineralization approach like MR agar, which are robust to the pleiotropic effect in our analysis.
Thank you.
Okay. So please note that people are asking questions in the chat. So please be sure to address all the questions that are being asked in the chat. Thank you.
All right. Thank you so much.
Thank you.
All right. So our next presenter is Zhang. And are you here? Okay, perfect. So yeah, take it away.
Thank you for the invitation. I'm a research assistant professor from Dr. Paul Robbins' lab at the University of Minnesota. I'm very happy to share with you one of our projects, which is Huixun, our novel senescence therapeutics that can reduce cellular senescence and extend health span. So it's bad that we all age, but the silver line is aging is malleable, and both genetic and pharmacological interventions have been shown to be able to extend health span and lifespan. So our aim is to develop pharmacological therapeutics that can extend health span by targeting the mechanisms of aging or hallmarks of aging. So the hallmark we have been focused on is cellular senescence. As you may know, with age, zombie-like senescent cells accumulate in various tissues and release detrimental inflammatory factors called senescence-associated secretory phenotype or SASP, eventually causing decline in tissue functions and driving aging and many age-related diseases. Two classes of senescence therapeutics have been developed to target senescent cells and demonstrate to improve aging phenotypes and extend health span. These include senolytics, a class of drugs able to selectively eliminate senescent cells, and xenomorphics, which suppress the detrimental SASPs without killing senescent cells. So to discover novel senescence therapeutics, we took a phenotype drug discovery approach by screening different libraries of natural products, FDA-approved drugs, and many others. The screening workflow is shown here. Proliferating cells can be induced senescent under different stress conditions, and then seeded in 96 well plates after drug treatment. Cells will stand with a fluorogenic dye based on the characteristic acid beta-gal activity, and then imaged with high content analyzer to evaluate the effects on senescence. So both senolytics and xenomorphics can be identified with this platform. So one of the libraries we screen is Huixun, a class of polysaccharides which can be found in many seaweeds. So we found that some sources of Huixun are senolytics, some are xenomorphics, some are more potent than the others. So we picked the most potent xenomorphic, which is Huixun-1 in the following studies. So here is the senescent staining of senescent mouse embryonic fibroblast cells. As the beta-gal senescent cells are stained in green and nuclei are stained in blue. As you can see, Huixun-1 can reduce green senescent cells, but not the cell viability. So therefore, it's a novel xenomorphic. Also, the xenomorphic effect is dose dependent. So we further confirm by qPCR analysis that Huixun-1 can reduce the gene expression of senescence and SASP factors in senescent MEF cells, such as P16, P21, IL-6, TNF-alpha, and MCP1. We further acutely treat naturally aging mice at 31 months old for only 5 days to see the therapeutic effect of Fucotin-1. As you can see, Fucotin-1 can reduce senescence in different tissues, especially in kidney. It also reduces P21 expression in freshly purified gamma delta T cells in the spleen. We also
giving you a 1 minute warning. Continue.
Yeah, we also evaluate its effect on health span in ERCS1 and mouse minus delta mice, an accelerated aging model. So mice at 9 to 10 weeks old were fed with Fucotin in chow for 6 weeks. We found that Fucotin-1 can suppress the overall aging symptoms, especially after 2 weeks of treatment. Also, the tissue analysis found that the reduction of senescence and the SASP factors in different tissues, especially in kidney. So in summary, from a focused drug screening of different sources of seaweed, we are very excited to find that Fucotin-1 from a particular seaweed is a normal xenomorphic. Fucotin-1 can reduce tissue senescence in naturally aging mice and accelerated aging mice, as well as extend health span. So we are currently performing RNA-seq analysis to investigate the mechanism of action or the potential targets of Fucotin-1. Hopefully we will publish the results soon. With that, I'd like to thank my advisor, Dr. Paul Robbins, and many collaborators, grants, as well as the organizers of this meeting. And thank you for your time and attention. I'm happy to take questions.
Thank you so much. Very interesting work. Did you notice any effect on overall longevity in addition to health span?
We didn't do any longevity study. It's just the health span. We just look at the overall aging symptoms, including like four grip strengths and kyphosis, or the muscle and brain aging. So we didn't look at the lifespan.
Sure.
Okay. Very interesting data. Congratulations. I wanted to ask you, did you see any effect on any mitochondrial components or proteins, or did you see an increase in oxidative phosphorylation or any other pathways?
No, we haven't looked at at a molecular level, especially mitochondria. Yeah, but we might look at that in the future. Yeah, thank you.
Okay, and did you see any effect in immune cells at all, or did you have you not studied that yet?
Yeah, we look at the immune cell in a fresh spleen, especially as gamma delta T cell, the subpopulation of T cells. So we see the reduction of P21 in these cells. So meaning it may reduce the senescence in these gamma delta T cells.
Okay, that's important for protective immunity. Yeah.
Yeah. Okay. Thank you.
Yeah, so there's a couple of questions came in the chat. If you don't mind, maybe responding in the chat, and we can all learn from those. Thank you so much.
Sure. Thank you so much.
All right. Our next presenter is Huixun Du.
Okay. I would like to first thank the organizer and the judges for choosing my talk. I'm Huixun Du from the lab. I'm a grad student from the lab of Dr. Dan Weiner at the Buck Institute. And today I'm really happy to present our interesting findings on substrate stiffness modulates senescent cells and SASPs. And just to a disclaimer is all the data currently I'm presenting will be published. So if you can just not save the pictures, will be great. Yeah, thank you. Okay, senescent cells with age, various stresses cause accumulation of senescent cells. And now we know that senescent cells and its secretory phenotype also contributes to many age-related diseases. Fibrosis, specifically to our interest, idiopathic pulmonary fibrosis is also very much of an aging disease, as its incident correlates significantly with the increased age. Recent papers have found the presence of senescence in the scar tissues of IPF samples. However, does IPF or tissue scarring also causes senescent cells manifestation as well as changes as SASP? That question remains unexplored. For that reason, we developed a hydrocell culture system where we use PDMS to coat on top of regular plastic plates. And we have two different tension or stiffness of gels. One is 2 kilopascal, mimicking the physiological state of lung tissue, and another fibrotic 50 kilopascal that mimics the fibrotic tissue in lung. We also compare these tensions to the traditional conventional plastic culture plates, as plastic has a tension that's much higher than either physiological or fibrotic. We then see the IMR90s. They are primary human lung fibroblasts on top of these different matrices, and we use doxorubicin to induce senescence. At the end experiments, we harvest the cells for senescent cell assays, and then we collect the media for SAS analysis using DIM mass spectrometry. So the very first interesting result that we got is after we culture cells on either 2 kilopascal or plastic, we actually see a drastic difference in the beta-gal positivity rates across various doses of doxorubicin. This means that cells that are cultured on physiological states, softer tissues, actually are attenuating their has less senescent cell formation. Then we analyze the SAS, where we also see a clear distinction of SAS secreted by senescent cells culture on either 2 kilopascal or plastic, as indicated in yellow and orange here, whereas the fibrotic 50 kind of lies in between. Then we plot out the secretomes, the differential secretomes of these senescent cells, and we see a general trend of tension-driven drives much more secretion from these cells. And when we summarize these upregulated proteins that are secreted by senescent cells, we actually see many of them are commonly shared between various comparisons. If we zoom in into the identities of these proteins, many of them end up being collagens, like the ECM components, or fibronectins, like filaments from the cytoskeletons. We then also ran pathway enrichment using IPA and GEO on these secreted proteins, and we can't see a consistent upregulation of fibrosis pathways, as well as integrin pathways, cytoskeleton-related pathway, and then adhesion-related pathways. What's interesting, though, is when we compare the fibrotic versus the physiological tensions, we actually see a unique group of terms coming up. These are immune activations. They are specifically coming up between these two comparisons. This suggests that fibrotic tissue culture environment itself could be more pro-inflammatory. And then we also send the cells for mRNA bulk MR sequencing, where the transcriptomic DEGs followed by pathway enrichment show that cells that are cultured on plastic, which is the harder surface, actually shows inhibition in MMPs. MMPs are groups of proteins that are proteases that degrades ECM components and essentially degrades collagens. What's very interesting along with that is we also see upregulation of many collagen proteins by the plastic conditions. And we also see an increased secretion, which I'm not showing here in mass spec data. So this together is actually suggesting that cells that culture on a harder surface actually are secreting more collagens, but making less MMPs, potentially making their environment even more pro-fibrotic. And when we send the cells for RNA sequencing, the cells that are treated with 100 nanomolar doxo, which we see this huge differences in the beta-gal staining. We ended up getting cells from the soft sites being upregulating a bunch of DNA damage checkpoints. So this essentially suggests that the cells that are cultured on soft stiffness might be upregulating their DNA damage checkpoints, preventing going into a senescence state. With that, I want to summarize. So we found that substrate stiffness modulates cellular senescence and the SAS profile. And we also see that high tension drives ECM fibrotic-related cell signaling pathways. And we also see that DNA damage checkpoints regulation could be a potential mechanism for tension modulator senescence, which we'll follow up further. With that, I want to thank especially to my mentor, and I want to thank the Schilling Lab for all the mass spec work, and I want to thank all my collaborators as well as the funding resources. Thank you very much for listening. I'm happy to take questions.
Thank you so much. Really amazing work there too. My question is related to can reversing this stiffness, or I guess reversing the disease affect the stiffness of the structure of the cells?
Could reverse the senescence process. So
Sorry,
So by using like antifibrotic mediators, could you reverse this process, you think?
Using antifibrotic mediators. So the whole sort of core theme of our project come about that sometimes these senolytics seems to work better in model organisms when the senescence manifestation hasn't been universal, but it seems to work not so well later on at a later stage. The reason being is the fibrosis tissue itself could be priming more senescence formation. So that for that reason, antifibrotic may not necessarily work, but obviously it needs more study. Yeah.
Okay. And what do you think is the relevance of the upregulation of the immune pathways and the inflammatory pathways?
What again? Sorry,
what do you think is the relevance of the upregulation of the inflammatory pathways?
Okay, of course, yeah. So that could potentially suggest that harder surface that during aging there is actually increase in many tissue stiffness. That is not necessary to the stage of a fibrosis. So that could very much be an underlying mechanism for the chronic inflammation that we're seeing.
Right. Okay.
Yeah. Okay. Thank you very much.
Well, thank you very much.
Thank you so much. All right. Our next presenter is Sam Collaudin.
Hi, everybody, and thanks for letting me present. Yeah, doing something at this conference. It's always a pleasure to participate and see all the very interesting panel discussion we had today. And so today I'm here to present not something about diabetes as usual, but something about telomeres and a new method that was developed inside Cell Environment Company by Radia M. Kasher. That is a long-standing researcher specialized in cytogenetics. And these new tools to analyze and improve clinical outcomes of patients with different age-related diseases, and especially today I'll talk about cancer. My disclosure, I'm an employee of Cell Environment and also a consultant at Kinexum. And here is the introduction. So as you all know well, I think telomeres are repeated DNA sequences that are associated with some protein complex, the shelterin, at the end of chromosome, and they have a very strong and important role in preserving genome stability and integrity. And the loss of these telomeres are known to activate some DNA damage response pathways and to result in chromosome end-to-end fusion and instability. And some of the diseases, and most of the diseases we are talking about these days at this conference, age-related one like cardiovascular, metabolic, diabetes, neurodegenerative, and cancers are all known to be correlated with impaired telomere length maintenance. That's the reason why it's very interesting to study and to be able to measure them very precisely and combine them with other biological markers to see how these different diseases are evolving, or they are impacting the bodies, and try to develop new personalized medicine. Let me present you what we are doing at Cell Environment. So Cell Environment is taking basically a very small drop of blood and able to analyze cell by cell a certain number of markers and combine them automatically from a system platform called ChromoDiag. So we are basically able to stain with a FISH method telomeres and centromeres. Here we talk only about telomeres, to measure the size of the telomere cell by cell, but also to see when you have telomere loss or doubling of the telomeres, for instance. And we combine that with genomic instability and chromosomal aberrations that we are able to measure at the same time to measure what we call a genomic instability score, and be able to use that in different type of diseases to do personalized medicine. I'll present here some of the results in one type of cohort we studied. We did a lot of work in different cohorts, and I could talk hours and hours about the work we did, but here is a very short presentation. I'll be happy to discuss more at another occasion. So we study a cohort of Hodgkin lymphoma patients and patients. We wanted to study the secondary cancer and the cardiovascular diseases in patients that went through treatments for this disease. We had access to 221 patients and age about 29 years, most of them in early stage, some in advanced, and we follow them for about 11.2 years as a median. And 10% of them develop a secondary cancer after the first treatment, and 26% of them develop a cardiovascular disease. And we wanted to study the evolution of the telomere length in this population and to see if it correlates with appearance of this complication or not. And here are some of the
1 minute warning, sorry.
Okay, yeah, no, cool. So on the left, you have patients that had no evidence of disease after the treatment. And basically, the telomeres doesn't really change. On the right, we separate the secondary cancer patient and the cardiovascular disease. And basically, you see on the left of each graphs that before or at initiation of the second cancer and the cardiovascular diseases, the telomere length was much shorter than at beginning or after the treatments. That's one of the things that we are able to measure. Just to give you another graph, it synthesized many work we did with different type of cohorts, measuring the telomere length with the age of the patients. In gray, you have the control patient, healthy patient, and basically we see with the biological age that goes on that the telomere is decreasing slowly. But in some cohorts, and I think it's very strong in the genetic disorder patients, for instance, you have a much shorter telomere length. You see also with patients in the light blue that has prenatal diseases that have a much smaller telomere length compared to healthy patients where we measure the cord umbilical telomere length. Some of them, we don't really see differences like patients with fertility, but when we combine that with chromosomal aberrations, then we really are able to identify the population and differentiate them from the control. So that's all for this short presentation, and I'll be happy to discuss more at other occasions. Thanks.
Thank you so much. Yeah, no, we really appreciate you coming to talk, and I know we wish everyone could present a lot longer, and I know you have a lot more that you'll be able to discuss with us. And so, my first question for you here would be, what do you think the biggest challenge would be in developing a tool that could be used or validated using telomere length or stability as a diagnostic tool?
Very good question. So the big, we have two challenges. One challenge is to find a good cutoff and see if a patient with a certain length have some risk of developing some age-related diseases. And I think to be able to manage and to solve the challenge, we need to combine telomere length with other biomarkers. And I think it's all the discussion we had today and previous, that the telomere length by itself is not enough, and we definitely need to combine that with other markers.
Okay, very good. So I have a question. Would it be more important for you to check specific immune cells in the peripheral blood than just whole blood, like to focus on lymphocytes, especially for cancer patients?
That's also a good question. So indeed, probably if we look at specific cancer cells, we'll see completely, we'll see different results. The goal of measuring, of this technology is really to measure the global health of the patients. And in cancer, for instance, here we are able to identify, to have some idea of of the development of future comorbidities by just measuring the blood. We can also measure the toxicity of the treatments I think it's also a very interesting part that we haven't worked on it yet. But basically, with the blood, we just get the overall health of the patient and not only the cancer cells that are definitely with a much shorter telomere length.
Congratulations.
Thanks.
Okay. And our final speaker this afternoon will be Lihuan Guan.
Hello, everyone. My name is Lihuan Guan, a PhD student from the University of Melbourne, Professor Andrea Meyer's group. It's my pleasure today to share my works. The title of my presentation is Unresolved Inflammation During Hospitalization is Associated with Post-Discharge Institutionalization and Mortality in Geriatric Rehabilitation in Patients, the Result Cohort. Okay. Okay. Sorry. Inflammation has been identified as one of the hallmarks of aging. Acute inflammation serves as a protective mechanism for the body against infections, injuries, and diseases. C-reactive protein and albumin are markers routinely used in clinical practice to assess systemic inflammation. CRP is an acute phase reactant and increases as a response to pro-inflammatory cytokines. And albumin is a negative acute phase protein, which is reduced during acute inflammatory process. After the acute phase, inflammation can be resolved or increased or become chronic and subsequently influence recovery from diseases. Both acute and chronic inflammation are associated with adverse health outcomes. In this study, we looked at the association of inflammatory markers, CRP and albumin during hospitalization with adverse health outcomes. The result cohort we used is a population of older and frail adults who had multimorbidity admitted to post-acute geriatric rehabilitation wards for restoring their functional capacity and preserving self-management ability. The median of age of our cohort is 83 years old, and 56% of patients were female. And we collected CRP and albumin data throughout the entire hospital stay, including acute and rehabilitation hospitalization, and calculated their median peak variation levels in each setting. To indicate the direction of change, increased CRP was defined if the last CRP measurement was greater than the median level before this last measurement. Albumin is the other way around. And we found that lower albumin during both hospitalizations was associated with higher mortality, and higher CRP during rehabilitation stay, but not acute hospitalization, was associated with higher mortality. And these findings demonstrates that high CRP and low albumin tested in post-acute rehabilitation might indicate unresolved and persistent inflammation from the acute hospital stay. And that this kind of inflammation can cause damage on tissues and organs, which will involve in the onset or progression of age-related diseases, and consequently increases the risk of mortality. Additionally, patients who could recover from high CRP or low albumin during rehabilitation had the best survival outcome, suggesting that those who could manage to overcome inflammation had a responsive immune system and are less likely to have their inflammation reoccurred or disease exacerbated. Furthermore, only increased CRP during rehabilitation was associated with institutionalization, indicating that patients who were discharged with unresolved inflammation were more likely to admit to a nursing home after discharge, regardless of the inflammation level per se. In conclusion, unresolved inflammation characterized by high CRP and low albumin in geriatric rehabilitation was associated with institutionalization and mortality, indicating ongoing disease activity leading to adverse health outcomes. And those results highlight the difference between an acute event and chronic conditions, and the importance of recovery from inflammation during rehabilitation. Thus, more attention should be paid to unresolved inflammation in geriatric rehabilitation patients. Yeah, thanks for your attention.
Thank you so much. Really interesting. As a clinician, when I have a patient that's elderly, that has low albumin, oftentimes they're very, they can be very malnourished. Chronically, it's a very, non-specific marker. I guess I'd like to know your comments on, if resolving, the underlying cause of hypoalbuminemia, or, what might be causing the inflammation. I guess CRP even more non-specific to a degree, but something like targeting a nutrition plan for an elderly patient. Would you expect to see increase in albumin? Would you expect to see, what would be the effects on their, risk of hospitalization? Did you guys look at that at all?
Okay, thanks for your attention. So sorry, thanks for your question. Yeah, so the low albumin is during the inflammatory process is due to when inflammation happens, the albumin synthesis rate will be decreased, and the albumin also can be has some leakage from intravascular space to extravascular space. So that's the like basic mechanism, like why low albumin happens during the inflammation. So regarding the increased albumin, if increased albumin can help improve the patient's health conditions. So though many literature states that albumin is actually just a biomarker of ill health condition. So it's just an indicator that this patient condition is not that good. So it will needs attention on what caused those health conditions, poor health conditions, rather than just the low albumin. So yeah, it's just a biomarker.
Sure. Yeah, definitely.
Okay, very nice, very interesting data. So how would you take this data further? Would you suggest that people just start on anti-inflammatory therapies for long term, or how would you proceed from now on?
Okay, yeah, so due to the limited time, actually I also did accuracy prediction ability analysis. So for these two markers, and also those different median aberration levels, those determinants has a moderate accuracy to predict the adverse outcome, like mortality. So it might be not that accurate to predict the post-discharge outcomes, but it's still we can see it's the rather than we focus on the inflammation during acute phase, we also need to pay attention to the left inflammation after acute state, because that matters more. So I think in the future studies, also the anti-inflammation interventions should be tested in those rehabilitation patients. So yeah, so what's the key point of this study is the unresolved inflammation after the acute phase. Yeah, so yeah, thanks for your question.
Okay, very good, very good.
All right, thank you so much, and thank you to all the presenters and everybody who sent in abstracts. It really enriches the conference, and we are grateful to learn from all of you.
And thank you very much for submitting such interesting work, all of you.